Okay, so what is mitosis? It is defined as the division of nucleus of parent cell into two nuclei. Okay, and each nucleus contains the same number of chromosomes and genetic content with the nucleus of the parent cells. And there are four phases: P-MET, prophase, metaphase, anaphase, and telophase. Okay, division of the nucleus into two nuclei. Okay, same number of chromosome, same genetic content as the parent cell. So it's like a photocopy machine. Okay, so there's a video clip for you to play. You can watch this, take your time and appreciate the video. Okay, also another video clip over here. So let's enter into the first phase of mitosis, that is prophase. Okay, so you need to know the chromosomal behavior in each phase. Okay, in these four phases, chromosomal behavior means what happened to the chromosome, what are the changes. So you must be able to draw the stages and to describe the events, especially the chromosomal behavior. So you see in prophase, what happened in the nucleus is that this is the nucleus, right, of the cell. So the chromatin, remember the term chromatin, the thread-like structure just now, it starts to shorten and thicken. So it's like growing fatter, short and thick to form chromosome structure that can be seen through a light microscope. So that means if it is not dividing, when the cells are not dividing, you cannot see the chromosome under the light microscope. So if you can see the chromosome under the light microscope, you know that the cell is now undergoing cell division or mitosis. Okay, it is in prophase. So the second point is that the chromosome is now able to be seen as two identical threads called the sister chromatids. Okay, so this is what we mean by sister chromatids. So initially when we draw chromosome, we just draw one like this, right? But now it has duplicated, replicated, so it's like a butterfly shape over here. Okay, so the right side and the right side and the left side, okay, they are sister to one another. Okay, so both sister chromatids, they are joined at the centromere. So you have to how to count the number of chromosomes in a cell. The tips is that you count the centromere. So 1, 2, 3, 4. So in this cell, okay, this cell contains 4 chromosomes. Okay, usually students, they don't know how to count the number of chromosomes. So very simple, you just count the centromere. Okay, so meaning this is considered one chromosome. Okay, but just that it is in the duplicated manner where it is now seen as the sister chromatids attached together at the centromere. So the nucleus membranes disintegrate. So you have to talk about this. This is the nucleus. So you have a nucleolus, remember that, in the nucleus, and you have the nuclear membrane. So what happened in prophase? The nuclear membrane disintegrates. So that's why when you draw the prophase, you have to draw dotted line over here because they are disintegrating. And the nucleolus cannot be seen now, so that's why some books they draw nucleolus but also dotted lines. Okay, with dotted lines. And the centrioles, so you see these centrioles, 90-degree you draw, okay, like a T lah. Okay, so this pair of centrioles moves to the opposite poles and spindle fiber starts to form. Opposite poles, that means this centriole is heading over this side, whereas this centriole is going to the opposite side. These two are opposite to one another now. Initially, maybe they are from one same place. Now they are migrating opposite to one another, and you get to see this spindle fiber forming. All right. Okay, so let's look at this picture, a very beautiful picture where you can see the faces over here. Prophase. So it's like a thread-like structure, chromatid becoming shorter and thicker over here. Right. Next is P mat MAT. So now after that, we're going to look at metaphase. So it's all in the middle. M, middle. Okay. After that, anaphase. Anaphase. So what are the chromosomal behaviors? They now have moved to the opposite pole. And in telophase, what happened? So we're going to look at that one by one.